All right, welcome everyone. Thank you so much for joining me this evening. This is another open hangout and an after show. This is the after show for the Dr. Wilson versus Nephilim free debate on modern day debates. So, if you're first time on this channel, the way that this works is that I am going to have a panel of people to talk a little bit about the debate before I open it up for a free for all. So I invited a couple of people to join me to discuss the arguments, and I invited Dr. Wilson, and hopefully he'll come. He said he may not be here, depending on how things go. So I would like to welcome the two guests that I have already, Dr. Dan, aka Creationist, and Atheist Junior. Welcome. Thank you guys so much for joining. Andrew Cummings, you may join in a little bit, but I'm going to keep it small for now before I open everything back up. Just because I don't want everything to be a shouting match and free fall in the beginning. I'll wait for that. So, Dr. Dan and Atheist Junior. Dr. Dan, let's start with you. What do you think?
Hi. So, okay. So, full disclosure, I was for the first time since last March. I went out for dinner. Like my wife and I went out for dinner, so I didn't watch the debate until just right at the end there, and I put it on 2x speed and went back to the beginning. So I only caught Dr. Wilson's opening, which was great. Hit a bunch of high notes that were awesome. The ribosomes, Hox genes, chromosome 2 fusion, bunch of great stuff. And then I got about halfway through Neff's opening, and I heard, just trying to remember all the wrong things, that mutations can't generate anatomical novelty, and chromosome 2 fusion isn't actually a fusion site. That's completely wrong. He claims there's like mission critical genes that span it, like multiple genes. That's wrong. There was a bunch of stuff like right off the bat that was just completely wrong. So par for the course.
Yeah. Atheist Junior, your thoughts?
Yeah, well, I did see the debate, or probably about 85% of it. And I thought Dr. Wilson did a good job at staying calm in the face of endless bullshit, which is commendable. And yeah, Nephilim, he had some strange arguments, like bringing up the Lenski long-term E. coli experiment and saying something like that the E. coli were eventually like released from the lab and died when that never happened.
I can tell you exactly what he's talking about there, but he's conflating two different things. And it makes it comes through the food processor as nonsense, but he's conflating two different findings from there. So the first thing is that within the E. coli, within the long-term E. coli experiment, they're adapting the E. coli to a very specific environment that is limited to one food source, glucose. Now, one of the lines evolved the ability to use citrate, but putting that aside, what they've done, because the cells are not using some of their metabolic pathways, their genomes have gotten smaller, because when you only have one food source, keeping the biochemical and genetic kind of machinery to like use those other food sources is a waste of energy. So you jettison those genes, you get rid of them. So if you were to put these E. coli back out into the environment against like wild type E. coli that have the full complement of metabolic genes in an environment where you have lots of food sources, the ones that are evolved in the experiment are going to be outcompeted by those ones out in nature. That's all that is. That's the first half. And then the second half of it is that there was a couple of papers recently about the citrate line, the one that evolved the ability to metabolize citrate and grows to a higher population density. The paper showed two things. One, higher mortality rate, which is just like exactly what you'd expect because they're at a higher population density. So like more of the cells die. That's just what happens. You have more competition, more death. And then the other side of that paper was that if you put them in a citrate-only line, they do not do well. They do fine in glucose. They do really well with glucose and citrate. If you have them with just citrate, that's not so good because they're not well adapted to a citrate-only environment. So what Nephilim Free did is he takes those two things and somehow puts them all together and says, the E. coli died. Like, I don't know.
Yeah, that's an interesting thing, talking about adaptation to environment, because like you said, like these E. coli, were thriving in the lab, but maybe they wouldn't do good put next to just normal E. coli. But then these E. coli would they can grow aerobically on citrate, which the others can't. So they're able to adapt to that environment. So it's context specific, So it just seems to me like he's describing adaptation.
I'm doing fine on land, but if you threw me in the ocean, I would die.
Right.
Yeah, exactly.
Yeah.
Yeah, and typically, as you mentioned, when one population evolves for a highly specified environment, once they leave that environment, their fitness will definitely go down for that. I have no idea what Nephilim 3 was talking about in the opening. He was talking about evolution being a Hindu ancient myth passed down from Plato or something like that. What on earth does that have to do with anything? And who cares? It just makes me scratch your brain and go, what on earth is he talking about?
Yeah, I missed the opening, but they spent a long time. Nephilim was getting into the semantics of morphology versus anatomy, and Dr. Wilson was trying to explain to him that bacteria don't have an anatomy, they don't have bones and internal organs, but he just couldn't get past that for some reason.
Yeah. I missed all of the open discussion. I literally caught basically from the start of Dr. Wilson's opening to halfway through Neff's opening. At the very end, I caught it. And then before I went back to watch it on Warp Speed, I caught just the very tail end of the Q&A, which I had no idea what anybody was talking about. I missed the question. And so it's just Neff doing his thing. We have to find a way to talk about Neff without calling him Neff because it's really weird. Because
yeah, I know. It makes me feel uncomfortable because I'm the real Neff. I'm Neff the Great. I'm the real Neff. There's a town in Pennsylvania. There's a town in Pennsylvania called Neffsville.
Oh wow!
There you go.
Are you the king of Neffsville?
Maybe one day. All right.
One of the questions.
Any particular points?
Well, I was going to say one of the questions at the end was good, that somebody asked him if he had hard evidence for intelligent design, and he kept rehashing this thing. It sounded like LPP wrote part of his script for this debate because he kept talking about how DNA has language syntax and all this thing. And he said that DNA has punctuation. And I'm thinking, even if you describe a nucleotide sequence as a code, it's only letters. What punctuation is there? So that was confusing for me.
He probably means stop codons as periods.
Oh, I see.
That's probably what he means. It's not punctuation in any sense of the word because it's biochemical interactions. It just has to do with the shapes of the molecules. It's not I don't know. I can't deal with the DNA is a DNA information. Fine. I don't care. DNA is a language. No, it's not. There's a field about linguistics and DNA. It's not a language. It's not a language.
Yeah. Yep. You don't see anybody but the ID people giving this argument.
Right. It's their argument, there's a code, there must be a coder. There's a law, there must be a lawgiver. That type of basic argument. So,
and I was watching the chat, and I had it on mute. I was periodically checking in, so I had the chat going, and I had the video on mute. And I saw that Dr. Wilson showed the random polypeptides abundant source of biochemical activity paper, which is yes, exactly. You can just do random polymerization and you get stuff. You don't need someone to decide what those things are. You just randomly throw either ribonucleotides or amino acids together and into polymers, and you get activity, you get stuff. And that's we've documented that. And so any argument that you need a designer for a protein to do so, no, you don't. You literally just do it randomly and you can get the activity.
Yeah. So there's one I kind of want to talk about is the chromosome number 2 argument. There's a lot of disinformation from the Yet crowd, specifically saying, I've heard some say that there's highly functional genes on the chromosome 2, therefore it can't be a fusion, and a bunch of other nonsense arguments related to that. Yep, pseudogene. So yeah, let's talk about what is actually there. What is that whole and
Yeah.
um,
Oh, I love that
basically
one.
all about.
That is
Yeah, you should do a
the.
creation myths to the that.
I'm actually so that one is something that I'm considering. I think I want to do the I think I want to do the Lenski E. coli die-off one pretty soon because that's coming up more commonly. You see that among a lot of people. But the chromosome 2 thing, it's that there's a pseudogene at the site there, and it is occasionally transcribed. It's not translated, but it's transcribed. And the reason that creationists claim it couldn't be a fusion because it spans the site is it has an alternate start site on the far side. So the whole pseudogene is on one side of the fusion site, and then on the far side of the end-to-end telomeres, there is an alternative start site. So you get transcription on one side moving away from the fusion site, and then occasionally a small, I don't know what the percentages are, but a small fraction of the time you get transcription across the fusion site. And the important thing about this pseudogene is that it's not it's part of a family of pseudogenes. So it's the DDX11L2 something. It's something D's and X's and L's, and I think there's an 11 in there. And it's a family of pseudogenes. And you know where all the other copies of those pseudogenes are in the human genome? Every other copy of that version of that pseudogene, they're all
On the other side
immediately adjacent to telomeres.
Yep.
Except for the one in the middle of chromosome 2.
Which is more evidence that it's actually from a fusion.
Yeah, the presence of that pseudogene next to the fusion site is actually stronger evidence of a fusion because those pseudogenes, that family of pseudogenes, the one in the middle of chromosome 2 is the only one not immediately adjacent to telomeres. But it's adjacent to the fusion site.
Yeah, and I believe that the human chromosome 2 has normally that chromosomes just have one centromere, but chromosome 2 there are remnants of a second centromere and vestigial telomeres that are just more evidence of this fusion.
Yep, exactly. Mm-hmm.
And of course, chimpanzees have basically a nearly identical DNA sequence for the human chromosome 2.
Oh yeah, and I saw it in Dr. Wilson's opening. He showed the picture where you line them up like human chromosome 2, chimp chromosome, like the banding patterns are all the same. It's the whole thing is top to bottom the same. You'd line the two up, and it's just there it is. There's chromosome 2, and that's a really great example of a confirmed prediction because you look at all the great apes, you have an extra pair of chromosomes compared to humans. So what biologists said when they figured this out is, oh, you know what? We should find a fusion in the human genome. We should be able to take those two chromosomes that don't correspond, like two gorilla chromosomes that don't correspond to any human chromosomes. We should be able to take those and fuse them together, and the end-to-end fusion is going to match the human chromosome that doesn't match to the other ape ones. And on the human side, it's human chromosome 2, doesn't have a match in chimps or gorillas. And you take the two chromosomes in chimps or gorillas that don't have a match in the human, when you do the karyotype and lay the chromosomes out, but you take those two chromosomes, smush them together, and it matches chromosome 2. That's it's a very specific prediction. And if that was wrong, that would have been a big problem for evolution. But
yeah, why would it have been a problem for evolution if this chromosome 2 fusion wasn't there?
Because it calls into question the similarity of the entire genome arrangement, right? So if you have, instead of having sets of chromosomes, right, like we have 46 chromosomes and 23 pairs, other great apes have 48 and 24 pairs. If it wasn't a one-to-one match, most of the chromosomes, and then a fusion, if it was just two completely different assemblages, right, patterns of which genes are with which other genes on chromosomes, then you kind of have to wonder, how did this one genome, right, if we're saying all these things share common ancestry, if there was no correlation in the organization at the chromosome level between the chimp genome and the human genome, now you kind of have to wonder, right? But we could say there is a correlation.
Right.
In fact, they're structurally basically identical, right? With the exception of the Y chromosome, because chimp Y chromosomes are weird because of their mating system. But other than that, they're like very specifically like boom, 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 straight down, top to bottom, each chromosome, except for the missing pair, right? So you say that the missing 2 in the chimp should correspond to the 1 in humans. They do. Prediction confirmed. Like,
All right, we have a really good question here. Dr. Dean, what is the best evidence for increasing genetic
Best
complexity that
evidence.
is metagenic?
Best
So,
evidence. I mean, that's a really best. There's a lot of good evidence. I don't know about best, right? So you've got the couple between 200 and 2,000 genes, probably somewhere in the middle there, between 1,000 and 1,500 is a better range. But you've got those some number of sites that are under positive selection between the common ancestor from chimps and humans and modern humans. So, like, that's a really good example that we've documented. In terms of, at least in terms of like brain complexity compared to the other great apes, humans have a bit of a leg up with our frontal lobes. So that's a good one. Let's see some other good ones. I mean, you could talk about like very specific functions. So you get like new biochemical functions, like the protein in HIV that inactivates a human protein called tetherin. You need to inactivate that protein if you're going to be a virus infecting our cells. And our tetherin's a little bit weird compared to like chimp tetherin, so you need a new mechanism to do it. So you get like new functions that way. Oh, you know what's a really good one? Here's a really good one, because this is a clear, specific de novo gene from a non-coding sequence, is the antifreeze protein that you find in some Arctic fish. Is they have this brand new protein that doesn't exist in any other lineages, but you can find a corresponding non-coding sequence in other lineages. And this non-coding sequence acquired, I don't know if it's transcribed in the sister groups, but it's not translated. And so in the Arctic fish, it acquires a start codon and a stop codon. It becomes translated. So through mutations, it becomes translatable, and it makes an antifreeze protein that allows them to survive in very cold water, where other fish would not be able to survive. That's like cut and dry, de novo gene, very specific function, clear example, and we've documented the entire evolutionary history. There's I don't know if that's the best, but that's a really good example.
Yeah, and I would just add that I think that there is almost some unnecessary complexity when it comes to genetics in a lot of different organisms, and I think that's just evidence of the sort of blind nature of evolution, and opposite evidence of there being a designer, because I think a hallmark of design is simplicity and efficiency, and not messy, overly complex stuff that just is sort of slapdash and happens to work.
Yeah, exactly. And we find evidence for that all over the place, both like morphologically and genetically, where we're using things that, like, you can see how it was just like we took this one thing and we're like, it'll be, it tweak it and it does this other thing, instead of having like a new thing that's specific to the function. You just end up tweaking things. And you find evidence for that littered throughout the genome. You find a protein family that does one thing, but like one member of that family has this other job. And like, I mean, okay, that works, but like, why not have something specialized for that new thing? Why do you have to take and kind of tweak some not quite optimal system for it?
So, anything else that Dr. Wilson or Neff said that caught your attention?
I still have to watch the whole thing. I'm going to. I just haven't. But I like I said, I really like the examples that Dr. Wilson brought out, because they were things like chromosome 2 and Hox genes. These are very specific. Like, they fulfill very specific evolutionary predictions, right? You're looking at all animal body plans or all bilateral body plans, and like, you can make a prediction. If they share a common ancestor with that body plan, then the same gene should be involved in organizing it. And turns out that's true. Missing a chromosome pair, we should find a fusion. That was true. Like, each of those examples hit a very specific prediction. And like, that's the name of the game, right? Make accurate predictions. Right. So I really enjoyed that opening. I thought that was really a good approach to do it.
Yeah, I agree. And I will give Yex credit. Yuck does make actual testable predictions. The problem is they've all failed miserably.
Yep.
Yeah.
So, yep.
Can I answer? I see a question in the chat that if I could just address real fast, because it's not on this topic. It's just from the flood counter thing.
Flood thing. Yeah, I'll highlight the comment. Was that the one?
This will be. I'll make this real quick because I have the answer here. Can Dan cite where he claims Dr. Brown makes the claims? Yeah, it was in his book. Hang on, let me get the name of the book for you. It was in his book in the beginning, Compelling Evidence for Creation and the Flood, and he says when he describes hydroplate theory, that these are his numbers, not mine. It's Walt Brown's numbers. 300 trillion 1 megaton H bombs. That's the energetic equivalent, according to the guy who came up with the idea. And then a physicist did the calculations on that. So Walt Brown provided the amount of energy, and then a different person, a physicist, converted that into temperature, which is just math. And what this other person said, what this physicist calculated, is if just 0.001%, that's 0.001% of the energy remained, then that would be 3,000 degrees Fahrenheit. So that's where you get the numbers.
Yeah, and since we're on the topic of the flood, might as well talk a little bit more about that. There is a really good paper from Walton University, I think, or Wheaton, whatever it is, called "The Physical Constraints in the Noachian Deluge." Have you ever heard of that paper?
Yep,
No.
I've seen that. Aaron Rod, I think, did a video about that.
Yeah, I actually downloaded
It was two Christians who wrote that paper, I believe.
Yeah, I downloaded the whole paper and I've read it, and it's a really interesting paper. They really go in depth with their calculations, and they really do a great job making sure all the calculations look good. And there's simply no way to get it to work.
Right.
And I think one of the points that I was saying the other night is that, as Maddie said on my channel, a global flood is not even possible from a meteorological standpoint. None of them have even tried to address how the storm system would form, what type of storm it would be, and all that type of stuff. So they literally can't even get their flood off the ground.
Yeah, and I just wanted to say I'm working on a video right now to show that when Kent says that he doesn't care what Walt Brown thinks or doesn't share beliefs with Walt Brown, that's a lie. And I noticed rewatching the debate that he said he's never met Walt Brown, and that is a flat out lie, because Kent has said before that he met Walt Brown in an airport one time, and they had a lunch date together where they talked about the flood and the canopy theory and all this stuff. So that's an outright lie.
Yep. All right, we have a couple more people to welcome, and I do apologize, but after this, my room is full for a little bit. We have Irrational Empiricist, aka You're Coming, the Church of Entropy, and Brenda. Welcome, everyone. I will first start with Andrew. What do you think of the debate and all of that type of stuff? What do you think overall? I know you did say you had a question for Dr. Dan.
Well, let's do debate stuff first.
Yeah, I can wait.
Yeah. Let's do the debate stuff. Okay. A Church of Entropy, what do you think?
Hey, you can call me Jen if you want. Church of Entropy is just the name of my channel. But thanks for having me. Can you hear me okay?
Yeah, I can. Thank you for
Nice to
joining
meet you,
us.
David. Thanks
It's
for
nice to meet you
hosting.
too. Oh, absolutely. It's my pleasure. Feel free to introduce yourself to the channel. I think it's your first time here.
Yeah, it is. I wanted to hop on because I found it real super interesting what was being discussed on the debate there. And nice to meet you, Dan, and Atheist Junior, Brenda, and Rational Empiricist. I liked what you said, Dan, about the citrate, quote unquote, evolution for the bacteria. I like the way you're going into that sort of. I like what you said. Sorry, I'm getting some
yeah, sorry.
That was gone now.
That's on my end.
As far as the debate, it was a little bit boring, unfortunately. I don't think there were any real slam dunks, and there really could have been. And overall, it was sort of drifting in and out, but some of it was a little bit okay, I guess. And they never really got to a point where it was clear that one person had an actual edge over the other, in my opinion, which made it a bit of a sort of, it fizzled out. And then I ended up asking the question at the end, how evolution via mutation predicts punctuated equilibrium. And I didn't really feel like the answer I got was very satisfying. So all in all, excited about talking about it, but the debate itself didn't find it super inspiring. And yeah, thanks for having me.
Well, Dr. Dan, you're an evolutionary biologist, so maybe we could talk about punctuated equilibrium and all that
We
without
can
your.
talk about punctuated equilibrium. Yeah, I actually, so common thing with punctuated equilibrium is that people think it was Stephen Jay Gould and who's the other guy? Who is the other guy? Niles Eldredge. Niles Eldredge in the 1970s came up with this idea, but that is actually not true. The person who actually first came up with the idea of punctuated equilibrium was none other than Charles Darwin himself in, I want to say, 1868. It was one of the later editions of Origin of Species. He changed part of how he explained some stuff, and he said, I forget the exact line, but it was something to the effect of the times during which adaptations occur, during which populations are changing, would be, he said, the way he wrote, he's a really good writer, but the language now is archaic, right? So it's, long, though long in years would be short relative to the periods of, during which stasis reigned or something, like whatever he said. But he basically said, like, yeah, you're going to have periods of rapid adaptation, but in between those, you're going to have longer periods of basically no change. And this was based on communications with other people in the 1860s following the publication of Origin of Species, and looking at, like, during the 1800s, we were like, oh, fossils, let's find all of the fossils now. And people were finding these and finding that you had periods where, like, there was rapid turnover and periods where there was not rapid turnover. And so Darwin incorporated this new information into his later editions, and basically said, yeah, the evidence points towards what we now call punctuated equilibrium.
Right. And of course, rapid speciation, of course, probably happens right after mass die-offs and extinctions,
Yeah.
correct?
Mass extinctions, anytime you open up a lot of niche space, so you can have mass extinctions, you can have rapid environmental change, which is part of what was going on in the Cambrian. So you didn't have a mass extinction right before the Cambrian period, but what you had is the environment was diversifying very rapidly. So you had more ecological niches available, so you get radiation. The other thing that can lead to rapid speciation is a type of mutation called a key innovation, which opens up a bunch of niches that were not previously available. So examples of that would be like pollen and flowering plants or seeds, right? Because now plants don't need water for reproduction directly to spread the sperm around. So now you're opening up a lot of niches. So we see an explosion of diversity among flowering plants at a very specific time in history, because they've now, through gaining a new morphological trait, have opened up a bunch of niche space for themselves, essentially. So those are the circumstances under which you tend to see the more rapid adaptation. Some more small-scale examples of that are like when a virus gets into a new host, you can document in their DNA, you can find rapid adaptive adaptation as they adapt to the new host compared to the old host.
Exactly. A great example of that being HIV has evolved from SIV, which is really fascinating.
HIV is a great example. Yeah.
Brenda, what do you think overall of the debate?
Sorry.
I just wanted to come in to talk to Dan because I haven't talked to him before, and I saw parts of the debate, but it's a tradition. It's just Neffy as usual, going through his usual shtick. I mean,
Yep.
I kind of have to agree with the lady. I don't know her name. Church
Jen.
of Entropy. Jen.
You can call me Church of Entropy if you want, but most people call me Jen.
That it was probably, I didn't see all of it, but yeah, it's kind of like uneventful, kind of boring. I don't know. So what do you think?
Semantics.
What do you think of why are we debating these people?
That's a great question. Why are we debating these people?
And by these people, I mean non-experts or non-informed people who are evangelizing, not debating, right? They're using the debate format as an opportunity to evangelize. And why not instead engage with a little bit more serious in this case creationists, but I mean serious opponents. That's what I'm trying to think
because
of.
they don't exist.
Yeah, like Dr. Jensen would not want to.
I'm just kidding.
Yeah, like Dr. Jensen would never debate Dr. Dan.
I didn't catch that. Sometimes you talk fast. I'm sorry, I didn't catch it.
I was saying like Dr. Jeanson would never dare debate Dr. Dan.
Okay.
So
Um,
If I could just say, like, I think the debates are still happening and they're still not going anywhere because both sides don't have the complete answer.
so
That
this is
might be a reason.
something that has been at the back of my mind, and I brought it up elsewhere, which is that I think that some of the opponents that we are talking to on internet through these venues are not people who have any kind of serious credentials or anything kind of serious to bring to the table.
No,
And
certainly.
I think we need to cut the cord.
I'm inclined to agree. My philosophy is kind of split on this, and it's one is that like you're not gonna shut Neff up. Like he's gonna be doing his thing. So either he's gonna do it unchallenged, or there can be somebody in the room that knows what they're talking about, like countering it in real time. Then like the latter is better. I used to come down on the don't give them the platform because the platform like gives them like more validity than they deserve, but like I don't think that argument works anymore when you could just go on YouTube and have like however many thousand people watching you for like like just some random person go on YouTube and a bunch of people watch. So it's like I don't think that give them a platform argument works anymore. So I think it's incumbent upon people who like know better to engage. But that being said, I think it's worth like limiting trying your best to like have the conversation with serious people and not like Neff.
Yeah, because that was a few years ago. That was the words that was in the atheist theist debate. I think was it was more like just by engage, and also in the global warming versus the climate deniers debates, which is simply engaging with these people is counterproductive. And I'm sort of echoing that, and I hear you because so I'm giving one side, but I'm fully aware of the other side. And I feel the tug of both sides, which is that there is a case to be made for engaging in all opponents, hopefully in an effective manner, because what really worries me is the even larger picture, because I see so many people who get caught up into anti-vaccine, and various other conspiracy theories that are extremely that are killing people. There are people for instance, I think we could probably attach a number to deaths due to conspiracy theories in Florida, right?
Yeah.
I think you could. Statistically, I think you could work out those numbers in not too long, and you can directly attribute them to vaccine denial, because there's a lot of old people there. And I just saw a report recently about an IT department, and all the IT, they were like, their entire department was decimated by people, by their department heads dying of COVID, right? Their department was decimated means significantly reduced in size, because there were several deaths within a highly technical field because of vaccine denial. So, and then bringing that back to the creation evolution debate, it's all part of a seamless thing where there is a loss of trust in authorities, particularly science, particularly any kind of association of government with science. And I don't know how to combat that, but I see it as potentially, even possibly causing real harm to the culture in the long run. Maybe I'm over exaggerating, Dan or somebody else. Maybe they can fill me in.
I'll let someone else, but I have two thoughts, but I'll let other people talk before
I was just gonna. Oh, sorry.
I agree. I think that people should use their voice and push back against pseudoscience wherever we can. But I think, this is YouTube. I think at the end of the day, the point of these debates is more for entertainment. And I found Dr. Dan's debate the other day extremely entertaining and enjoyable.
Okay.
Scientific debates are that are done in a professional sense, I think are more going to be boring, but these can be pretty enjoyable to watch most of the time. But I don't know if it's counterproductive. I think that and there are people who are on the other side who do get convinced by people like us who debate. They might not say it, but I do think it happens. And I think if I can convince one person not to believe some of this stuff, I think it's worth it.
Yeah, I would just commenting on what Dr. Dan said. I think I agree, and I think that these people, creationists and other pseudoscience advocates, may not know what they're talking about, but one thing's for sure: they do know how to attract those views and those subscriptions. So I think that is one thing to keep in mind.
And something that came up to my mind, which was I saw a video a few days ago from the UK of a UK journalist being chased down the streets by an angry mob because of they were 5G deniers.
Oh,
I mean, no, I'm not kidding. He literally had to go behind a barricade to escape from them.
The journalist said that 5G wasn't
because the 5G people in the UK, it's really bad, and they just blame journalists and anybody connected with official governments and stuff like that for they think that they are being their DNA is being altered by 5G. That's what they believe.
Wow. Yeah, for some reason, there's a lot of flat earthers in the UK as well. I'm not sure why exactly that is.
I'm not sure either. I think I have derailed, and I should probably apologize for that. I brought my own issues to the table. I'm sorry, but
yeah.
Well, I've got an answer for you. The solution to bad science is just better science, and so the people who are complaining about people believing crazy things, they need to look at what they believe and make sure that it actually accords with science properly so that the false beliefs will be displaced in the process. I don't think the evolutionary people are presenting a strong enough argument, basically.
Until the angry mobs come and drag you onto the pile of books from the library and chant.
I thought you said you wanted
gods.
to stop derailing the stream.
Well, so I have two thoughts on the broader question of like what's the is it valuable and what's the point? And I think I've come down to like it is, even if it's like for entertainment and frankly like kind of silly. Like the other night I debated Kent Hovind about the Ark, which is like a dumb topic. Like no, there was not a literal 500-foot wooden boat. Like that didn't happen, right? Like it didn't happen.
I saw that.
Like,
that was good.
Like why do that? Because the two reasons. One, if you allow people like Kent or Neffy or whoever to say like these stupid scientists are all lying to you, yada yada yada, then what you're doing is you're basically just promoting a distrust in like our ability to acquire knowledge and have confidence in knowledge, which is bad. We as a society should like value processes that allow us to determine what is true and not true. So undermining that just in general is bad, even if the specific topic you're talking about is dumb. And then the second thing is by training people to like believe stupid conspiracy theories, like that Bill Gates is trying to mind control people with a vaccine, or like 5G is something or other, like, or the earth is flat, like something harmless, like the earth is flat. When you train people to believe these things by teaching them and like rationalizing how they could be true, you're teaching people basically to disconnect the BS detector part of their brain, and that makes them susceptible to other BS, right? So, and that's bad because like if you believe in a flat earth, you're more susceptible to also believe that vaccines are going more likely to hurt you than a virus. And like it doesn't matter at the end of the day if anyone believes in a flat earth. There's no real societal cost to that directly. But if it makes more people into anti-vaxxers because they're like primed to believe misinformation, then that's really bad, and that has a tangible cost for everybody, right? So
Yeah.
that's why it's, I think, incumbent to like have these conversations, even if the topic is silly and doesn't matter, right? Because the point is to like exercise that critical thinking muscle and get everybody to do that.
Yeah, and I think there's actually evidence of this because Kent Hovind is an anti-vaxxer, and if you look in the comment section, Kent posted the debate on his channel. There are so many people in there that got pissed off by the vaccine comment, you know? So lots of his viewers are anti-vaxxers as well.
Not only that, but Kent Hovind also promoted poisonous medicine, I think, to cure cancer, which is totally
Latrial?
disgusting. But anyway, I kind of wanted to kind of direct this thing, but direct the conversation back to the flood a little bit. I alluded to this paper earlier, the physical constraints of the Noachian deluge. Really great calculations in here. I know this is a 1983 paper, and but creation in science really hasn't gone too far. The biggest things they developed since then were the catastrophic plate tectonics and a couple other things that makes absolutely no sense. But here are some of the calculations that they do. Scientific creationists maintain that biblical legends can be explained on sound science. Since miracles cannot be predicted, they have no place in the science curriculum. And I like what they say in bold. We do not consider in this paper the even more difficult problem of disposing of the water as the flood recedes. So they're not even good. So that also is a big problem. So rainfall method, here we go. How much atmosphere would be needed? 840 times higher than it is now. 3,500 degrees Celsius, 6,400 degrees Fahrenheit. Here are the calculations. If anyone wants a copy of this paper, please email me and I will gladly send them to you.
Yeah, the math on that doesn't work. It's fun to play with those numbers. They don't work at all.
Oh yeah, and if you add in accelerated radioactive decay and catastrophic plate tectonics, that makes the problem even worse.
Yeah, my favorite with that one is the who was it? I think it was Jordan actually pointed out in a debate that he was in that had to do with Age of Earth. He pointed out that if you accept the young Earth and you buy radioactive decay, and even if you come up with a solution for the heat problem, which the people who like are all about accelerated nuclear decay, the rate team, they say there's no solution. It needs to be exotic solutions, aka miracles. He said, but Jordan pointed out that if you are going to take that idea seriously, putting aside all the other problems, there's food is bananas are radioactive because there's potassium in them, and some fraction of potassium is radioactive. The extra radioactivity from food on the Ark would kill everybody on the Ark.
Yeah, and actually, Jordan and I actually did a follow-up on that, and I think it'll be fun just to play a little bit of a portion of that clip, the heat problem. This is from my video response. Jordan and I did a video response to one of SFT's videos. I'm trying to see if I can pull this up. If not, well, I'll just do without it. Share.
I also just want to. While you're pulling that up, I also just want to go back to something Jen said a few minutes ago about that like people need to make better arguments. I think at some point it ends up being like an identity thing. Like you really buy into stuff, and it's not something you believe because of evidence. It's something you believe because it's part of your identity to believe it. And at that point, there's no evidence that can change that person's mind. And like Neffy in the debate, I caught just a little bit of the Q&A, and someone asked him, like what evidence would change your mind? And he said the same evidence that would convince me. It was something about like, little green aliens or something. And it's like, yeah, there's nothing that's going to convince Neffy because it's not about facts. It's about like part of his identity is believing this thing. And if you're asking someone to accept new evidence that would require them to completely refashion their identity, they're not going to do it. So yeah, it's not. I don't think it's as simple as just better arguments, more facts, more information. I think it's more complicated than that, and that makes me very sad.
Yeah.
Yeah. Okay, I found exactly what I wanted.
Death by bananas.
Yep. You're,
Dan, I think the one solution to that, I have a lot of experience debating flat earthers, actually.
Yeah.
And
hold on, before you go there, let me just
Go
play
ahead.
this part real
Go
quick.
ahead. You're
Sorry.
right. I thought you were not going to play it, so that's why. Go ahead. Sorry. I apologize.
On it, but don't worry, I did. So I'm going to share the math now, and we'll walk through it together, and we'll find out if, in fact, 0.01% is okie dokie, right?
Okay.
So, yeah. So let's just take the banana. I didn't say bananas. I don't know why he's all about bananas, but let's just go with bananas since he's all angry about them.
Yeah,
right?
it looks like I guess you could say that Frog Mat is bananas about bananas.
Yeah, since he's bananas about bananas, we'll just stick with them. Okay, so let's do the math. Bananas have a little bit over 400 milligrams of potassium, and 0.01 whatever percent of that is potassium 40. So that's 49 micrograms of potassium 40. So we need to ask the question, how dangerous is that? Well, this is the formula for radioactive activity. That's the activity. This is the number of atoms in your sample, and that's its decay constant. This is that term in half-life, which people are more familiar with. In order to know the number of atoms, you do this formula. I'm not going to walk it through. You can pause it if you want to read every variable. If you just put those two together, you get this formula. And so if you crunch the numbers with today's decay rates, the activity of a banana is about 13.1 becquerels. A becquerel means 1 decay per second. So in your banana on your counter, about 13 potassium 40 atoms are decaying every second, which is not dangerous at all. But rates we're talking about accelerated decay rates, right? So this is what it is. I had one more slide. This is the amount of energy that is decayed. Every decay releases 1.3 mega electron volts, which is just a unit of energy, just like joules. So that's it in joules. Your bananas put out 2.7 picowatts of energy. Not a whole lot. But anyway, we're accelerating.
I don't know if it's the same for you guys, but the video's a little weird for me.
Yes, me too.
It's
Yeah,
kinda
it's choppy.
robotic.
It's choppy. Yes.
David, are we okay? Uh-oh, did we lose?
I'm here.
We're still here, so we haven't... Have we... Oh, we're back.
Yeah,
Matt, are we okay?
I think it's his bandwidth.
Uh-oh.
Yeah, I agree. I agree. Probably bandwidth.
I can't even share videos on StreamYard. It makes my audio robot like that, so I don't even try. But in the interim, I will add. I wanted to add what you said about the identity of young Earth creationists. We do know that there are people who are young Earth creationists who, come out of it and you start accepting evolution. Like obviously David is an example, but it's when you have a YouTube channel and you're a public figure making these arguments, especially for a decade like Neff, it's a lot harder to sort of admit, oh, I was wrong this whole time,
It's
Yes, it's
Uh-oh.
You're still roboting.
Uh-oh. Oh, no,
sorry.
we lost David. Oh, no.
He's probably refreshing the page.
Yeah.
Dan, can I run by you what my issue is
Please.
with
What
evolution?
issue with what?
with evolution? Not an
Great, let's
issue with
do
you.
it.
I've got no problems with you. You actually sound like a super smart, spoken guy.
I appreciate that. I figured it wasn't personal. I was just I was wondering what topic. I figured it wasn't personal. That was
I
I
agree.
just always run through things logically. So when I try to run through evolution logically, this is where I get to. So the evolution happens because of random mutations, which cause greater fecundity in the offspring, which over time overtakes the entire group
that
due
is
to this
one
higher
mechanism. Yes,
fecundity. So is there another mechanism on top of that?
there's a lot of other mechanisms. Yeah, so that's a really good summary of natural selection, right? It's you have heritable differences in the DNA, and the ones that confer a reproductive advantage become more frequent over generations. Just by virtue of having more offspring, you just are represented. I should say, by virtue of having more offspring than average, right? The frequency goes up over generations. So that's one mechanism, but that's far from the only mechanism. Another mechanism is mutations just on their own. You know, you could take mutations as a separate, like as their own mechanism, right? So you've got natural selection changes the frequency of different alleles or genotypes. Natural selection or mutations generate new ones. That's important. You also have genetic drift, which is the same as natural selection, except instead of being based on reproductive output, it's random. So you can have random fluctuations in the frequencies of traits. So this is going to be something either that doesn't have like a major effect on your reproductive output, or it could be a situation where maybe there's like a natural disaster and whoever survives the flood, it's random. It's not based on who's, camouflaged to hide from predators best, right? It's just
perhaps the best example for this would be the city that was destroyed by a volcano. That Greek one, I forget what it was called.
It was Vesuvius. It was the mountain, and Pompeii, right? So
there you go. Yeah, so like a Pompeii type situation. It was just basically like, see ya.
Yeah, and it's right, and you have like whatever fraction of the population happened to be in a boat at the time, survived, right? And that's like random with respect to most genes in the genome, so the fluctuations change randomly, right? It could also just be a product of like sampling error when it comes to reproduction, that you get random fluctuations. And then you get things like recombination, gene flow, horizontal gene transfer, where you're mixing and matching to get new combinations between individuals or between populations or even between species. So you can shuffle around genetic components between different entities. I use the word entities. It's kind of a catch-all for populations, individuals, whatever, just between different entities. You can shuffle around and you can generate novelty that way. And that often opens up a lot of new traits. And an example of that is the placenta, where the reason the placenta can develop, one of the linchpins in that process is when the embryo initially implants in the uterine wall, the mother actually responds to that as though it's an infection and there's an inflammatory response there. And in marsupials, the reason they're born so underdeveloped is they don't have a way to suppress that inflammatory response. So marsupials are born very underdeveloped and then finish developing in the pouch. But for eutherian mammals, like placental mammals, we actually stole a gene from a retrovirus that invaded our own genomes. It was kind of a stealth mode protein, which you can imagine is useful for a virus, right, to kind of hide from the immune system. And when you express this protein at the point of implantation, it suppresses that inflammatory response. So the mother's uterus is unable to evict the embryo. So it can grow much larger and develop a placenta, and you can have robust development in utero. And that one change facilitates this completely new way of doing embryonic development. And it's because mammals acquired a gene from a virus that invaded their own DNA. So like that's an example of a completely different mechanism that gets you some really new and cool stuff.
Cool. Well, that last one actually makes a fair bit of sense to me. I think I would largely agree with that. Where it falls apart for me is that I look at the overall map of all the animals in all of history, and it appears to be punctuated equilibrium, i.e. the same species for a while, and then something new starts, and then it's that same new thing for a while. And then so that is not what the slow evolution of random mutations predicts, right? The slow evolution from random mutations predicts a continuous deformation from one species to the next.
Yeah, so it did initially. So that's like the 1860s version. And then that changed starting in the 20th century, because when Darwin first proposed the idea of, he was started off like straight up gradualism, like basically linear change as you're describing. By the time you get into the 1900s, people are basically saying not so much, still on the gradualism side of the spectrum, but not as much. Because we started to have an understanding of genetics, and you had Gregor Mendel and then Thomas Hunt Morgan and a bunch of other people, and you had the association between genes and chromosomes, and we realized that genes were kind of quantum units, where you could have like variant A or variant B, or in some cases, variant C, D, E, many variants, and it wasn't a smooth gradient from one thing to another. It was kind of discrete classes for many traits. And when you have that, you necessarily can't have a smooth gradient as traits change. You either have state A or you have state B as the population changes. And then once you get into the middle of the 20th century, that's when we kind of started to figure out like a bunch of these other mechanisms. So you had the understanding of like DNA at the molecular level and like biochemically, like how mutations actually work. You had the understanding that there are small-scale mutations and large-scale mutations, and those large-scale mutations often facilitate large-scale changes, whereas the small-scale mutations often involve fine-tuning once you've adapted, once you're able to use a resource. That often involves a big change, and then small changes to kind of refine your ability to do that, like the within population adaptation. And then you have other mechanisms that are even more recent, so things like epigenetics and phenotypic plasticity, which is like last 40 to 50 years kind of stuff. And all of these strengthen the case for the punctuated equilibrium side of things, because for whether you're talking about gradualism or punctuated equilibrium, you need an underlying molecular mechanism. And for a long time, we had the underlying molecular mechanism for gradualism. Single base point mutations, gradual changes over time. But now, starting 50 to 60 years ago, we now have a much more robust understanding of the underlying like molecular genetic mechanisms that would lead to the punctuated equilibrium pattern that we see in the fossil record.
Yeah, I guess my issue is
something?
how do you use the theory to predict whether it's a large transition or a small one? The theory doesn't appear to be able to do that. I'm not saying your explanation wasn't great. It was very insightful. I'm just trying to show you what my issue is.
I'll give you a specific example. Would that be okay?
Well, I'm talking about how do you use the model. All I'm asking is how you use the model to predict whether it's going to be a large or a small transition. I don't think it can do that.
So I don't think you can necessarily look like in a forward-facing way and say like, okay, I have this population of organisms, and in order for them to, I think in the future, they're going to change in XYZ ways, and that's going to be based on a certain genetic mechanism. I don't think you can do that without like doing some underlying work in the lab. So we can, for example, we can look at, in the lab, we can do experimental evolution to evolve bacteria to be resistant to specific antibiotics. Basically to say, okay, here's the susceptible bacteria, here's the drug we want to use to treat them. How are they going to evolve under that selective pressure to beat that drug? And that allows us to kind of predict in the field, right, when we're actually using the medication, what mutations we expect to see, and then we can have a plan B for when those mutations occur. And this has actually happened a couple times where we do the experimental evolution in the lab, we document what pathways are most likely, and then a couple years later, you could actually see those mutations occurring out in nature, and now we're like ready for it. So we can say, okay, we did the experiments in the lab, we documented that it's these specific, very small scale mutations. Now let's go out and like see, do we actually see those in nature? And the answer is yes. On the large scale side of things, we can look at like fossils, and we can say, okay, at this point in the past, we have a big transition in morphology. So if we can get the genomes of the organisms on kind of either side of that transition, the ones that have the ancestral state and the ones that have the new stuff when it appears in the fossil record, we can look at the descendants of those and say there should be some really big genetic differences there, like major genetic differences. And an example of that is full genome duplications among vertebrates. So invertebrates have one cluster of, it's actually genes that Dr. Wilson mentioned in the debate, about one cluster of Hox genes. When you get to vertebrates, you get two clusters of Hox genes. So you have a full genome duplication. And this actually facilitates like developmental complexity. And then when you get to a group called the gnathostomes, which are the fish that have jaws, gnatho is jaw and stome is mouth, so gnathostome just means jaw mouth. And you look at all the gnathostomes, we're gnathostomes, 4 Hox clusters. So you got a second full genome duplication. And that makes perfect sense. That's exactly what we would expect based on the fossil record, because we look at the invertebrates, the jawless fish, and then everybody else with jaws, right? And we could say, okay, those 2 steps from invertebrate to vertebrate, and then from jawless to having jaws, those are significant, because it's not just jaws, there's other stuff that's more complex. Those are significant increases in complexity that appear to have happened relatively suddenly in the fossil record. And we predict, therefore, that should correlate with like very stark differences in the genomes among those different organisms. And what we see is evidence for 2 sets of full genome duplications leading to greater developmental complexity. So like, that's a large scale example where we can make a specific prediction, and then we find the genetic evidence that was like, yep, that's what we expected to find.
I'm afraid that you
can I
can't
add
provide
something real
evidence
quick?
for your axiom.
Is it okay if I respond to her question?
Yeah, go ahead, because I'm not sure I'm understanding.
Yeah, real quick.
Okay.
Respond, and then there's a really good question in Cyber Chat, and then after that question, my internet connection is getting kind of spotty. So after this and after the question in Cyber Chat, I think we'll start shutting down just because my internet connection is kind of poor right now.
Okay. Well, I think one way that, I could be wrong, but one way that you might be able to predict if there's going to be a lot of random changes in the alleles of or versus stasis, or a small amount of changes, is if you look at the species, because we know that certain species like insects don't really care about the partner they have when they're mating. But non-random mating happens if you look at birds, mice, fish, primates. Like with birds, we know that they select their mate based on the vivid coloration or how complex their songs are. And humans tend to mate with individuals who resemble us phenotypically. So the sort of non-random mating with individuals that are like us usually will result in genotype frequencies that are, in favor of homozygotes, or, you have similar alleles versus animals that just kind of mate with whoever's there, where you have a lot more randomness, I would assume, in the sequences of DNA. So I guess that would be my response.
All right,
That's
so
another
here's a really
method.
good question. Yeah, can Dr. Dan expound on how the ERVs roles in evolution? That is a fun question.
That's a super fun question. I love ERVs. So ERVs are endogenous retroviruses. They are, let's start with, what's a retrovirus? A retrovirus is a virus that has an RNA genome and it converts it to DNA. So that process is reverse transcription. So they're called retroviruses because they do it backwards relative to how cellular life does it, which is DNA to RNA. So these retroviruses, they integrate into your genome. So like they convert themselves to DNA and then cut your thank you, that's nice. You cut their host DNA and stick the retroviral DNA in there. And this is a problem for the host because like you can't get rid of it once it's in your DNA. So like HIV right now, if you have access to like the really good HIV medication right now, it's a chronic manageable condition. It's not like going to make you sick. You can maintain a completely zero viral load. But you can't get rid of the ones that are integrated into your DNA, right? They're still there. Now, most of those are evolutionary dead ends because they integrate into your DNA, and most of your DNA doesn't make it into your kids, right? Only your germline DNA has the potential to make it into your kids. Well, sometimes retroviruses infect the germline, and if they get passed on generation to generation, now you have vertical transmission of this viral DNA in your own genome. Well, those viruses, they can mutate. And not only that, but we have enzymes that actually recognize viral DNA and go boop, 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 and just mutate the crap out of it. So what happens is you're left with things called endogenous retroviruses that have integrated into your DNA and then mutated and got stuck, and they can't cut themselves back out and reactivate. About 8% of the human genome is endogenous retrovirus, and that's compared to about 1.5% that actually codes for like proteins, like human genes, exons specifically. So we're more dead retrovirus than we are human gene in our genome, which is super cool. This matters because those retroviral sequences are biochemically and genetically active. They can serve as the raw materials for variation in the host organism. So the big example of this is the thing I mentioned earlier with the protein, the genes that are expressed during development that facilitate placental development early in embryonic development. The other thing that they do is they have their own promoters, so the regions that turn genes on and off. So we've actually co-opted some retroviral promoters to be part of the human gene regulatory network. So we use some of their promoters for our genes now. And then the third thing that's really useful is we use them because we want to, they're viruses, right? So we want to inactivate them. So we've actually adapted some of our antiviral defenses to recognize the dead retroviruses as targets for inactivation. So we just do things like methylation, which prevents them from being expressed. Because if you express them, they're viruses, so they could potentially cause problems. So the short version, so that was a lot, and I apologize for the length of that answer, but the short version is it's viral organisms inserting their DNA into our DNA, and by our, I mean animal DNA, and then the animals have that additional source of raw materials that can be co-opted to do something interesting down the road.
Because they're highly flexible design.
Sorry, David, I didn't catch that.
One of our friends' favorite arguments: ERVs are highly functional, therefore they're designed.
Yeah, they're not highly functional. There's a couple that have been co-opted, but for the most part, they're so degenerate they don't actually do anything. Like the vast majority of them, there are thousands upon thousands upon thousands of them. I think there's over 100,000 ERV sites in the human genome, and the vast majority are so degraded. The only activity they have is maybe they have a little bit of low-level transcription, but for the most part, they don't do anything. It's the ones that would, in order to be functional, they would have to be mostly intact. And we can identify that, and we can look at the ones that are mostly intact, and they're not functional. Most of them don't do anything. Like I said, there's a couple where we've co-opted either the genes through exaptation, or the promoters. We've based we've adapted to where they landed in our genome and kind of co-opted them to regulate our own stuff. But those are the exceptions, not the rules. The vast majority, they don't even have enough activity to be functional.
Hey, David.
Yes.
Oh, okay.
I think Andrew's cutting out too. Or is it just me?
Am I cutting
Yeah,
out?
he's cutting out.
Uh-oh.
Yep. Can everyone hear me?
I can hear you. Yes.
Yeah, I can hear you.
My internet.
David, you're just
Good time.
really bad.
Sad face.
Dan, are you familiar?
Can you guys hear me all right? Am I still on?
We can hear you, but you're robot.
Now I can. Yeah, now I can. hear
Okay,
you.
well, let's just put it this way. This will be a good place to stop because we've been going for a little bit over an hour, and my internet connection is kind of dying. So, good place to stop. Great discussion tonight. Thank you guys so much for joining. I do apologize for my spotty internet connection. But feel free to go around the room at the end if you have any last words to go around.
No, all good. I just I would love to Jen, Brenda, Atheist Junior, we've talked before, and Andrew, we've obviously talked, and David, we talk all the time. But anyone wants to continue this conversation down the road with David on this channel, my house, whatever, I'm happy to talk. So by all means, Jen especially, I'd love to keep talking, but I don't think tonight's the time. But I would love to continue this conversation at some point.
Sounds great. Nice to meet everybody. Thanks for hosting, David.
Nice to meet you
And
too.
I'll sub to your channel. I get it's
Thank you.
creation.
And
It's just so I'm sure.
Yep. And
Yep.
if you enjoy these open hangouts, if you enjoy these types of streams, please hit that subscribe button, bell notification down below so you can stay up to date when I'm doing these streams. I try to do them at least once every other week. And a couple of things I'm working on. I am working on a response to accelerated Christian education, which is all kinds of bad. And finally, I have an interview that I'm trying to schedule with Jordan from Reason to Doubt. We're talking about one of my personal favorite things, which is nuclear power. So don't forget to hit subscribe so you can know when that is going to happen. It should be later this month or next month, but that's going to be a lot of fun.
Yeah, and if I can say real quick, this coming Wednesday, I'm going to be talking to Jackson Wheat on my channel. And after that, I know Dr. Dan, if you want to come on the channel for a little interview Q&A, we can schedule that. I would love
Would
for you
love
to
to.
come on. And I've just released on my Patreon a video just responding to some viewer comments. So that's going to be premiering tomorrow. And then I'm also working on, as always, working on a video about Kent Hovind and Walt Brown. So that's going to be coming out soon as well.
Cool.
All right. Thank you, guys.
Thank you.
Thanks,
Subscribe
David.
to these people.
Too bad.
Subscribe to
Bob.
Dr. Dan. Subscribe to Dr. Subscribe to Dr. Dan. Subscribe to Atheist Junior. Subscribe to Church of Entropy. Lots of fun. This has been a great discussion. Great time, guys. Have a great day. Bye.